All right, let's dive into a genetic mystery that's had scientists scratching their heads for decades. It's really a story about evolution itself, a puzzle about how brand new code gets written into our DNA. And the main clue? Well, believe it or not, it comes from a handful of very particular fish.
So, at the heart of all this is a really fundamental question. How does evolution create something totally new? I mean, we know genes change and adapt, but where does the brand new stuff in our DNA actually come from? Specifically, how do new bits of code get jammed into genes that already exist?
To really get to the bottom of this, you need to meet our main character, the intron. Now, the easiest way to think about this is to imagine your DNA is a movie script. You've got the parts that actually make it into the final film. Those are called exons, and they build proteins. But introns? They're like the deleted scenes. They're right there in the original script, but they get snipped out before the final movie gets made.
Now, you might think adding a deleted scene to a script is no big deal, but in genetics, it's the exact opposite. Gaining a brand new intron is an exceptionally rare event, especially in vertebrates like us. It just almost never happens, which makes every single time we see it a major clue in understanding how our genomes are built.
Which, of course, brings us to the core mystery. If this is so incredibly rare, what on earth could be powerful enough to stitch a completely new piece of DNA into a gene that's been perfectly stable for millions of years?
To answer that, well, we have to go underwater. The first real clues to solving this puzzle didn't come from where you'd expect. They came from the genomes of a few peculiar fish.
Okay, so the genes at the center of this whole thing are the melanocortin receptor genes, or MC receptors for short. Now, these genes are famous throughout the animal kingdom for being incredibly stable. They're clean, they're simple, and they almost always come in one solid, unbroken block of code. They just don't do introns.
This here is what that stability looks like. See those long, solid blocks for humans and zebrafish? That's one continuous piece of genetic code. It's read from start to finish, 0 interruptions. This is the normal expected version. This is how it's supposed to be.
But then, scientists found this. In 4 specific types of fish, including the pufferfish, that clean, solid gene was suddenly, well, it was broken up. You see those white gaps interrupting the block? Those are the new introns. They're the deleted scenes that just popped up where they absolutely shouldn't be. And this wasn't just some random fluke, it was a clear pattern.
And this was so important because it wasn't random. It was a lead. So like any good detective, the researchers started following the evidence, connecting all the genetic dots to figure out what in the world was going on.
You know, when they mapped out exactly where these new introns appeared, the mystery just got deeper. The insertions weren't sloppy. They were unbelievably precise. This diagram shows the gene, and each of those colored arrows, it points to the exact location of a new intron. Just look at how they all line up on the same spots across different genes. This was targeted, not accidental.
And the evidence they found was just so specific. Get this. First, one of these new introns appeared in the exact same spot.
In 2 different but still closely related genes. I mean, the odds of that happening by chance they're basically 0. But it gets even crazier.
This same ridiculously precise pattern of insertions, it was found repeated perfectly across 4 totally different species of fish. At this point, it's not a clue anymore. It's a smoking gun. It's a genetic fingerprint pointing to one single cause. This discovery demanded an answer. I mean, what on earth did these 4 very different fish have in common that could cause such a specific and rare genetic event? And that question led to the big aha moment, the clue that tied it all together, their compacted genomes.
So let's just ask the question directly. What's the one shared trait between the pufferfish, the medaka, the stickleback, and their relatives that makes them different from most other fish, like, say, the zebrafish?
And the answer is the size of their genetic blueprint. While a fish like the zebrafish has this big, sprawling genome, these 4 species have genomes that are tiny. We're talking severely compacted. They've just shed massive amounts of DNA over their evolution.
This process is actually called genome compaction, and you can think of it like a frantic, huge spring cleaning. The species is under some kind of evolutionary pressure to be more efficient, so it rapidly just throws out huge chunks of DNA it thinks it doesn't need, shrinking its entire genetic blueprint way, way down.
And this realization led to a pretty radical new idea.
What if this whole process of shrinking the genome, this genetic spring cleaning, wasn't just about throwing things away? What if, in the process, it could accidentally build something brand new?
So the theory goes something like this. First, the genome goes through this rapid compaction. And this is not a gentle process. It puts the DNA itself under immense physical stress. That stress can cause the DNA strands to literally snap and break. Then, the cell's emergency repair systems rush in to stitch everything back together. But in that mad dash, mistakes happen. A loose piece of non-coding DNA gets accidentally stitched into the middle of a gene, creating a brand new intron.
And this isn't just a wild guess. The authors of the study that found this connection concluded that, and I'm quoting here, genome compaction is now believed to be a new hallmark that promotes intron insertions. This was a huge statement, basically proposing a whole new way for evolution to work.
So why does this matter so much? Well, first, it's a totally new way for evolution to create something. It directly links a massive genome-wide event, like shrinking an entire library, to a tiny specific change, like adding one sentence to a single book. It shows that physical stress, something we think of as destructive, can actually be a creative force. And maybe most importantly, researchers found this wasn't a one-off. They found the same pattern in other totally unrelated genes in these fish, which proves this is a real widespread thing.
Which leaves us with a pretty provocative final thought. You know, we usually think of evolution as this process of adding complexity, building things up. But this research suggests that the drive for simplicity, that pressure to shrink and become more efficient, might actually be one of evolution's most powerful and maybe accidental creative tools. A messy spring cleaning that ends up redecorating the house in a completely new way.